This is the Professor, CBMD PhD, and today's Thursday, July 30th, 2026. It is 5:10 and 52 seconds a.m. Eastern Standard Time. And today's research, Super Agers. So let's hop to it here. Scientists discover a compound that could supercharge aging, muscle repair. Scientists may have created a super hepatogrowth factor that helps aging muscles repair themselves more powerfully. Date July 24, 2026. Source, Kyushu University. A sulfur-based compound called LASSS appears to protect and supercharge a key protein involved in repairing damaged muscle. The discovery could eventually lead to new ways to slow muscle loss and preserve strength as people age. Skeletal muscle often begins to deteriorate relatively early in the aging process. Over time, this can cause loss of strength, increased scarring, fat buildup within muscle tissue, and a decline in fast-twitch fibers, which support rapid and powerful movements. When muscle tissue is injured or exposed to mechanical stimulation, hepatogrowth factor is released. It then attaches to c-Met receptors on satellite cells, the stem cells responsible for maintaining and repairing skeletal muscle. This signal brings the cell out of their inactive state, allowing them to multiply, mature, and help rebuild damaged muscle fibers. Aging can disrupt this repair system. Previous research from the team found that hepatocyte growth factor can undergo a chemical modification known as nitration. During this process, a nitro group is added to the two locations on the protein, Y198 and Y250. These sites are located in the same region hepatocyte growth factor uses to connect with the c-Met. After nitration occurs, hepatocyte growth factor can no longer attach effectively to the receptor. The researchers compared the damaged protein to a rusted key that no longer fits its lock. This loss of function may be one of the underlying causes of muscle wasting and reduced regeneration in older adults. Hepatocyte growth factor is not necessarily missing as we age. Rather, it can be chemically altered after it is made. That led us to wonder whether a compound with strong antioxidant capacity might protect hepatocyte growth factor either by preventing nitration or by compensating for the functional loss it causes. The researchers and scientific investigators investigated two compounds with strong antioxidant properties, glutathione trisulfide and lipoic acid trisulfide. Both are trisulfides, a class of molecules containing three sulfur atoms connected in sequence. These compounds have attracted growing interest in pharmaceutical research because of their distinctive sulfur chemistry and their ability to participate in redox reactions. Early experiments showed that both glutathione trisulfide and lipoic trisulfide reduced nitration at the Y198 and Y25 sites on hepatocyte growth factor. However, neither compound fully restored the protein's ability to bind to its receptor. The researchers then increased the molar ratio of hepatocyte growth factor to trisulfide, moving from 1 to 4,000 to 1 to 8,000. The higher concentration produced an unexpected result. When hepatocyte growth factor was mixed with lipoic trisulfide, its ability to bind to c-Met rose to more than twice that of untreated hepatocyte growth factor. The protein also became more resistant to the loss of function caused by nitration, particularly at Y198. This improvement was seen only with lipoic trisulfide, and glutathione trisulfide did not produce the same effect. This exceeded our expectations. We knew trisulfides had diverse biological functions, but we never expected that simply mixing hepatocyte growth factor with lipoic trisulfide would produce such a striking effect. What this tells us is that lipoic trisulfide does more than simply neutralize reactive molecules. It may interact directly with hepatocyte growth factor and induce a subtle structural change, creating an enhanced super hepatocyte growth factor from that binds c-Met more strongly and resists nitration. The findings suggest that lipoic trisulfide may directly alter the structure of hepatocyte growth factor in a beneficial way. Rather than acting only as an antioxidant, the compound may create a more active form of the protein that connects more strongly with its receptor while resisting chemical damage. To determine whether the protective effect could also occur in living tissue, the team tested lipoic trisulfide in mice with muscle atrophy caused by tail suspension. Mice treated with lipoic trisulfide before the procedure had significantly lower levels of nitration than untreated mice. Once again, glutathione trisulfide did not provide measurable protection. These results indicate that the beneficial effects of lipoic trisulfide are not limited to laboratory experiments involving isolated proteins. However, additional studies involving aging animals will be required to determine whether lipoic trisulfide is safe and effective in vivo. The discovery could support the development of new approaches for maintaining muscle repair during aging, extended bed rest, and other conditions that involve long periods of inactivity. The researchers believe the effects of lipoic trisulfide on hepatocyte growth factor may apply across multiple species, including humans and companion animals such as cats and dogs. In the future, the approach could potentially help people maintain strength, independence, quality of life, and a longer, healthy lifespan as they grow older. And this videography was created for educational purposes only. My code name is Will. I'm Professor Christopher Brunet, leader of research scientist, ufologist, psychic, computer technician, computer programmer, philanthropist, writer, mathematician, artist, photographer, film producer, chef, animal, and nature lover. My passion is world peace. Have a great day. This is the Professor, CBMDP.